Basic idea being that the mitochondria and chloroplasts look like little cells inside your cells, and so that's probably what they are. That at some point in history, there were big cells engulfing little cells, and one of them happened to get lucky and engulf a little cell that was really good at cell respiration, was really good at turning food into energy, and helped both the big cell and the little cell. And the big cell helped the little cell by taking in lots of little critters that could be digested. And so, the evidence here is pretty overwhelming. The double membrane from it being taken in, the circular DNA that looks like bacterial DNA, their own ribosomes that look like bacterial ribosomes, and the fact that they reproduce just like bacteria on their own and not directed by the cell is pretty hard evidence that this theory is our best explanation. So that is sort of the end of the cell parts section of the chapter, which is probably good. I think we've got enough cell parts in our brain now. How did the little bags of water thing go? Feel pretty confident? No? Okay. Well, we'll see how it went. It grades automatically. So moving on now to our next phase, our next part of the chapter, which is all about how things get in and out of the cell. You know about cell parts now. Now we got to talk about how stuff moves into and out of cells. We call that transport. In other words, how things are transported into and out of cells. In order to talk about transport in cells, because cells are basically bags of water, they're bags of water floating in water, we're going to have to talk a little bit about how things behave in water, or how they behave in solutions. So, I think you know some of this stuff from your basic science education. What do we call stuff that is dissolved in a liquid? We usually say solute. I think that's just to distinguish it from this. It's not a solute. But yeah, solute. Solute is the stuff that's dissolved in a liquid. So if I were to take a jug of water and make some Kool-Aid, pretend this is water, not vinegar. That'd be going to be a really gross Kool-Aid salad dressing. Kool-Aid vinaigrette. Yeah, so let's say I make some Kool-Aid, right? I put the powder and the sugar in there, the red powder and all the sugar that's supposed to go in there. I make Kool-Aid. The powder and the sugar are the solutes, right? Well, what do we call the water? What do you call this? Yeah. Good. It's called the solvent. That's the liquid that the stuff is dissolved in. Okay. What do we call the two together? That's the solution. Simple enough. Those are easy. Solute, solvent, solution. But now we need to find a way to talk about how much solute there is. Not just how much there is, but how much there is per amount of solvent. So let's say I make 2 gallons of Kool-Aid. And in this gallon of Kool-Aid, I put, a gallon of water, and then I put the recommended, the 1 packet of Kool-Aid stuff, and then however much sugar they say to add, like a cup of sugar, right? And I mix it all up. In this one, I put 2 packets of the Kool-Aid stuff and 4 cups of sugar, and shake it up like so much that all the sugar won't even dissolve in there. What's the difference? What word can we use for the difference between this Kool-Aid and this Kool-Aid? The one I used, the regular amount, and the one I used a whole bunch more. What do we say? That this one is more what than this one? No, solubility is sort of a measure of how much solute can go into a solvent, and that's going to be the same on both. The word is concentration. Excellent, excellent. That's a word that's important. It's one of those words like density that it's not an absolute figure as far as how much solute there is. It's how much solute there is per volume, right? And it's usually expressed in mass per volume. Like how many grams of sugar and Kool-Aid I put into how much volume of water. You know, maybe it's 100 grams per liter. That's a measure of concentration. Concentration is how much stuff there is dissolved in a particular volume of fluid. Okay, we're gonna do a little experiment. All you have to do, I'm gonna light this candle on fire. And your job is gonna be to raise your hand when you smell it and keep that hand up. Good? Raise your hand when you smell it. Yeah.
Whoa, I haven't seen one of those since 1973.
Raise your hand when you smell it. Whether it's the match or the candle. The masks don't help. McKinley's got it. I think it's like brown sugar and sweet potato. Yeah. Sweet. Who makes a candle that smells like sweet potatoes?
Monsters.
Well, oh no, I like sweet potatoes. I just don't want my candle to smell. Notice the pattern. I'm not quite there yet. I'm feeling left out. Right, you smell it? Hazen? No?
It could just be kind of congested right now.
Look at him, he can smell it.
I'm too proud. What's the general pattern? Yeah, it went from where the candle is outward in basically a circle. If we were in 3 dimensions, if there were people sitting above and below you, that would be weird, and it would be a sphere, right? It goes outward from where it started. So we talked about what smell really is, right? That you're actually taking pieces of the thing you're smelling into your nose and they're hitting those receptor proteins. Yeah, you can't unknow that now. But that's important to the concept we're about to talk about. What I basically did was concentrate a whole bunch of those smelly molecules right next to McKinley's head. And it turns out any molecules that you dissolve in a fluid, and air counts as a fluid, just like water does, any molecules you dissolve in a fluid are automatically moving randomly, which means they're moving there's thousands of them, and they're moving in all different directions at random. So if you put a bunch of them in one spot, what it's going to appear that they're doing is moving outward, when really they're moving all different directions. But basically, that sets up what we call the laws of diffusion. In diffusion, the way we can kind of define that using the terms we know now is when random motion of dissolved molecules, they have to be dissolved, they have to be in solution, but when random motion causes them to move from, well, let's use our concentration term. What to what? They move from what to what concentration? Fill in the blanks. Random motion, I concentrated a bunch next to McKinley, and then they moved from what to what concentration?
A to B.
A concentration to B concentration? That doesn't make sense. Concentration is either high or low, right? It's high to low. It's as simple as that. Don't overthink it. Stuff moves from high to low concentration. Until there is no more high and low concentration, right? Until they're equally concentrated throughout the room. Until wherever I am in the room, if I pull an air sample, it's got the same number of maple, brown sugars, whatever that is, molecules in it per volume. We're probably about there. Some of them are sliding under the door. McKinley's getting a higher dose just because that thing keeps throwing molecules into the air. I'll help him out here. Shut it down.
Candle is so medium.
Medium?
Yeah, it's not a good smell, but it's not a bad smell. It's very medium.
It's just like my grandma's.
That's nice.
It reminds me of my grandma.
I know.
That's nice. Happy fall.
Yeah.
Decorative gourd season. All right.
I
Yeah.
have these really good smelling fall wax.
Good smelling fall what?
Like wax melter. Have you ever like a wax melter? It's like
Wow.
and when you put it in the thing and it just heats up the wax.
It's so good.
A wax melter. All right. It's like a safety candle. I learned something today. Yes, Brady. Shh, can't hear Brady.
I thought that, like, you know, whenever I would go into like a craft store or something like Halloween, and you just get that instant whiff of pumpkin spice.
Yes.
That made me not like pumpkin
Just
spice.
like, yes, me too. My wife and kids love everything pumpkin spice, and I
I don't like the taste of it.
disagree strongly. We could probably start a culture war right here, right now, with forming up on sides of pumpkin spice versus not. Anyway,
Springboro divide.
here's the deal. This matters to cells because it turns out there are molecules that can simply just diffuse right through the cell membrane. They gotta be little molecules. Typically that means gases and water. Most gases and water can pretty much always diffuse across the cell membrane. So the way that we're going to demonstrate that we understand how all this stuff works, and it's about to get harder than just diffusion, diffusion's easy stuff, is what we call beaker problems. But I'm going to cut that off for now. We'll do our first beaker problems tomorrow. I'd like to talk about what I call the big lie, which is this: that cell basically in an organism like you does not exist. That cell that has, you know, one or two of each organelle type, and it's all balanced, and it's a teaching tool. It's a tool to teach you cell parts. That's not what your cells look like. Your cells have a buttload of whatever they need to do the job that they do, and not much else. They are generally overloaded with a couple of kinds of organelles and pretty skimpy on the others. And so this is just our third way. We did an analogy, and we did simple language with the bags of water activity. This is the final thing. This is like how I find out if you really know what these cell parts do. Because what this is a list of human cell types, 5 human cell types, real ones. And I'm describing on each of them exactly what their job is. And you have to figure out which cell parts they should have a lot of based on what job they do. In order to do that, you're gonna have to understand what the cell parts do, right? How they get jobs done. I don't know how it's gonna go, we'll see. But again, I'm gonna insist that you do your own work here so I know that you understand the stuff. This looks easier than it is. It is multiple choice. Okay, there's just a list of cell parts. On this one, no guarantees. You might use one cell part more than once. You might use one not at all. Okay, so keep that in mind. It's not going to be as neat and clean as the plain bag of water one. And again, you should expect it to be graded closely for correct. There are correct answers here. You need to find them. Go ahead and get started. That'll do it. There's only like 7 minutes left.
I don't know.